Hi, class. It's Dr. Lindner. Okay, so we've spoken about some of the properties of muscle, the functions of muscle, different types of contraction of muscle. We spoke about muscle histology, the endomysium, the perimysium, and the epimysium. We spoke about origins, insertions, innervations. Now we're going to talk about some terms that are really important to understand muscle physiology or how muscles contract. Now, in a previous video, we had mentioned atrophy and hypertrophy. We said atrophy is when muscles aren't being used, or if you have nerve damage going to a muscle and it can't be used, the muscle's going to waste away. That's muscle atrophy. When the brain is sending messages down the spinal cord into the nerves that go to the muscles, and the muscles are being activated through exercises, those muscles can hypertrophy. They can thicken and get larger. So muscle cells are stimulated to contract by motor neurons, somatic motor neurons. So if this is the brain, and here's the spinal cord, and let's say here's a nerve, and that nerve is going to muscle, there is a highway of communication where the brain can send down information down the spinal cord to the nerve, to the muscle. Well, that's called motor. But the motor receives information and it can send feedback of information from the muscle to the nerve up to the brain, and that's called sensory information. Maybe it's providing information about tension, maybe it's providing information about the speed of the contraction. So you have a motor neuron and you have a sensory neuron. The neuron that makes the muscle contract, which is voluntary, is called a somatic motor neuron. The exact attachment of this nerve to the muscle is called the neuromuscular junction, the NMJ, the neuromuscular junction. That's where neurons make contact with the skeletal muscle. This is an example of a neuromuscular junction. So here is a somatic motor neuron, and the somatic motor neuron has many branches that come off of it. They're called axon collaterals. And if we look at the end of an axon collateral, there are many end bulbs that are located there. Now, at the end of the end bulb, if you remember from a previous video, at the very end, remember you had dendrites, cell body, and you had an axon. So you had the dendrites, the cell body, and the axon. And at the very end of the axon, there was a swollen tip. And that swollen tip is called a synaptic end bulb. And what we have that exists here are neurotransmitters, which are chemical messengers. And the neurotransmitter here that's very important, that's going to interact with muscle, is called acetylcholine, ACH, acetylcholine, ACH, acetylcholine. That's the neurotransmitters that are located in the synaptic bulb, and somehow these have to move in this direction to stimulate the muscle to make it contract. And what we have happening is that calcium, which hangs out here, calcium has to influx inward. calcium moves inward, which ends up being a messenger that tells the acetylcholine to open up and to release its contents in this space. This gap, or this space between the neuron and the muscle, right? It's called the neuromuscular junction. But this gap, or this space right in here, is called a synaptic cleft. The synaptic cleft is the space where acetylcholine needs to be released to cross the rivers here to get back to land, which is the muscle. Okay, I'm going to go over this a few times, but this is just laying down some general framework of information. Okay, now here is the muscle fibers. Remember, you have a bunch of muscle fibers surrounded by an endomysium and a perimysium. So we call these myofibrils. These are the muscle fibers or muscle fibrils. Notice there's lots of mitochondria here. Lots of mitochondria. And these little myofibrils, they're made up of actin and myosin. These are contractile muscle proteins. And they're microscopic. They hang out in here. You can see, if you look deep to this blue meshwork, what we have here is going to be something that looks like, I'm going to kind of like oversimplify this. Let me just change the color here. We're going to have these different proteins. And the red one looks thicker, and the black one looks thinner. So myosin is thick. It's the red one. And the actin is the black one. And the myosin is going to have these extensions coming off of them that are called myosin heads. Okay, so this little structure between actin and myosin, the way these interact with one another, they call that a sarcomere. And this sarcomere is happening all in here. All of this stuff that I'm circling are microscopic sarcomeres, and you can see it like these overlapping red and blue lines underneath the blue meshwork. Okay? All right, we have a T-tubule right here. This T-tubule, the T stands for transverse tubule, stands for transverse, because it runs transversely to everything else that's going left to right. That one's going up and down. So we have a T-tubule, which is where the electrical conduction travels down. And then what connects to it is this blue meshwork called the sarcoplasmic reticulum. All of this blue spider-like matrix-like looking thing, that's the sarcoplasmic reticulum, and it's really important because what happens there is that is where calcium is stored. So we have calcium storage in this blue stuff. And the sarcoplasmic reticulum, its job is to either hold on to the calcium or to release it. So somehow calcium is involved in muscle contraction. We're going to see how that happens. So what we see on the top left, the NMJ, the neuromuscular junction, here's an axon collateral, right? Here's the axon. It looks like this right here could be a Schwann cell. You have an axon collateral, and right at the very end, right at the end, we have a synaptic end bulb, right? That's this thing. That has these little neurotransmitters that's going to connect somehow to the muscle, right? So at the very, very end, we have these end bulbs. Now, where the nerve attaches to the muscle, they call it the motor end plate. It's called the motor end plate. Okay, that looks like an egg, like the egg yolk broke. Okay, that's going to be where the nerve and the muscle make contact. Okay, now what we're doing is we're going to take this right here, we're going to blow it up and look at it to the right. So again, we have the axon terminal. We can see the nerve impulses traveling down the axon. It's going to make it right to the very end where we have the end bulb. And in the end bulb, we have these little circular vesicles. Okay, those little circular vesicles, are filled with a neurotransmitter. The neurotransmitter is called acetylcholine. And acetylcholine is always excitatory, always excitatory at the neuromuscular junction. And remember what I said earlier is that the acetylcholine has to find a way to make it to the muscle. It has to cross this gap or this space. And that gap or that space is called the synaptic cleft. So you could see that right in here, it looks like these vesicles are opening and releasing its contents to cross the synaptic cleft. And what gave it the message to do that was calcium. Calcium influxes. It goes in, sends a message and says, hey, acetylcholine, you can do your job. You can cross the synaptic cleft and you can bind to the motor end plate of the muscle. So now we're taking this that's boxed in and we're going to look at it over here. So we can see in these circles, we can see the ACH is released from the synaptic vesicle. It's crossing the synaptic cleft. What made that happen, remember, was calcium. Calcium influxed in and it told the acetylcholine to cross the synaptic cleft. And on the motor end plate, on the motor side, this is the muscle side, this is the nerve side. On the muscle side, we have these green receptors. So the ACH, the acetylcholine, binds to this receptor. And this receptor is like a key. The key goes in the lock, it opens the door, and what rushes in is sodium. Na is sodium. And whenever sodium rushes into a cell, it creates excitation or depolarization. So when sodium rushes in, you see the red arrow looks like a lightning bolt or electricity. That's when a muscle action potential is produced. Okay? This area right here, this invagination, that invagination, that's the T tubule. That's this thing right here. So the action potential rushes down that T tubule. Okay, so let's look at it again in a different illustration and talk it out. So here's the neuron on the upper left. There's a motor neuron. These are the dendrites. This is the cell body. Here's our axon. And at the very, very end of the axon, look at that box. That's where we have the neuromuscular junction. Everything in here, these are the myofibrils, which is the actin and the myosin. You can see that in here. And somehow these are going to interact with one another. So right up here on the top right, this looks like the neuromuscular junction. This right here is the synaptic end bulb. And you can see those little dots are going to be acetylcholine. This is the muscle side. This is the nerve side. The acetylcholine crosses this space that's called the synaptic cleft. It's going to bind to receptors. That's where that sodium rushed in, moved down the T tubule. That action potential, the T tubule, if we go back here a second, where does the T tubule contact? The T tubule, look what touches it. You got this buttressing of the sarcoplasmic reticulum makes contact with the transverse tubule. So the transverse tubule sends down this electrical action potential, transmits it into the sarcoplasmic reticulum. And what does the sarcoplasmic reticulum store? Calcium. So now here, the T tubule is carrying the action potential to the sarcoplasmic reticulum, which stores calcium. So the calcium is going to be released. When it's released, when the sarcoplasmic reticulum releases the calcium, look what's deep to it. Those muscle fibers, which is what we have up here, that actin and myosin. So somehow calcium is going to act on this. So the calcium is released, acting on the thick and thin filaments, the actin and myosin, and a muscle contraction will take place. And the term contraction means that there's tension being generated. So when I come back, we're going to review the parts of the sarcomere and try and understand what calcium's role is in the contraction.